Greetings, people. We meet once again. Still, we're talking about the patterns of inheritance. Earlier on, we talked about three of them that falls under types of dominance. We talked about complete dominance, incomplete dominance, and codominance. Now, I'm here again to assist you in understanding polygenic inheritance, which is the fourth one in this that we have. Here you will need a calculator. There is a calculation that we will do. So you can see that in paper 2, we now have the first calculation. We now have the first calculation if I exclude calculating the percent of a heterozygous or homozygous dominant. So here you will need a calculator. Bring a calculator closer to you. You will do some calculations along the way. Let's understand this one, polygenic inheritance. What is polygenic inheritance? We say polygenic inheritance is a mechanism of inheritance in which a genetic characteristic is controlled by many pairs of genes instead of a single pair of genes. What are we saying here? We are saying, people, this is a type of inheritance whereby a characteristic is controlled by many pairs of genes instead of one pair of genes. Take note of the part that I've highlighted there. I've highlighted many pairs of genes. If you look at the term poly means many. If you can go back to grade 11 when we are talking about what? Polygenic. So when we are talking about what? Polypeptides. So we say poly means many. So what is happening here? You can claim it by saying poly. So there's what? There's many pairs of genes. But let me assist you to understand this in another way. When we say it's an inheritance in which a genetic characteristic is controlled by many pairs of genes, according to our own understanding, we understand that a characteristic will be controlled by a single pair. That's what we understand, me. We understand that a characteristic will be controlled by a single pair. Now here we are saying, when we talk about polygenic inheritance, we're saying this is an inheritance whereby a characteristic is now controlled by what? By many pairs. So you will find that here it's not controlled by a single pair, but it is controlled by many pairs. So you can see now that we have one pair, we have two, we have three. So we have many pairs of genes controlling just only one characteristic. That is polygenic inheritance. So make sure that you understand it in that simpler way. It's just a definition, people. For us to understand this, having a build up to the calculation that we'll have, we must understand it from this slide. Make sure that you understand everything here. This is the heart of understanding polygenic inheritance for you to master the calculation that we'll have later on. We say, people, there are polygenes that are there in the polygenic inheritance. These polygenes, we say these are group of genes that add value on a particular phenotypic characteristic. It's just a definition of polygenes. But then we categorize these polygenes into two types or into two categories. We have additive genes or additive alleles and non-additive alleles. Please make sure that you understand the difference between the two, between additive allele and the non-additive allele. Okay, fine. When we talk about an additive allele, people, we say this is an allele that add a set of amount to the phenotype. Or one can say it add value to the phenotype of an individual. But then when we talk about non-additive allele, now we say it is an allele that does not add anything, it does not add value on the phenotype of the individual. Okay, fine. I want you to understand the difference here. An additive allele, people are saying, once there is an additive allele, it means there's an amount that will be added on the phenotype. But once there is a non-additive allele, there's no amount that will be added. I would like to focus on the first one, the additive allele. So we're saying here, there is a value that will be added. So we are saying additive allele, it adds what? Value on the phenotype. How do we see that this is an additive allele? Make sure that you cram this. An additive allele, people, it is what? It is a dominant allele. We will see it by what? By a dominant allele. Now, let's understand the difference between a dominant gene and a recessive gene in a simpler way, in terms of how we write them, how we show them on paper. We said a dominant allele, we show it using what? Using a capital letter. So meaning, people, we will see that it is an additive allele if it is what? It is a capital letter. Once there is a capital letter, it means that is what? That is an additive allele. That additive allele, it will do what? It will add value, or it will add a set of amount into the phenotype. But a non-additive is not a capital letter. I hope you get me clearly here. Once kwaba a capital letter, it means there's a set of amount that has been added. So you have to understand what an additive allele will add how many, or will add which amount. Let's go on to the next slide. Oh, before we go to the next slide, the last bullet there I said, the overall phenotype is equal to the sum of the additive allele in the polygene set. So how do we get a phenotype of an individual? Firstly, we'll be given a phenotype. Maybe they will give us a genotype, then a phenotype. Then for us to get the phenotype of an individual, sizo add the already given phenotype, see add the with the additive alleles that we have there. Let's see it here. Let's use this example. In this example, people, I said milk yield is controlled by many polygenes. Let us say the genotype, here is your genotype. The genotype gives milk yield of 4,000 liters. With this genotype, you will get how many milk production? 4,000 liters. Now they say each dominant gene, a dominant gene in a simpler way, it is what? An additive allele. What does an additive allele? It adds value on the phenotype of the individual. What are they saying? They're saying each dominant gene adds 400 liters to the yield. So meaning, if you put a dominant allele here, a dominant gene, there will be 400 liters that will be added. That 400 liters is added on top of what? On top of the 4,000 liters. Let's go on. This means that the genotype here is our new genotype now. Do you see that we have a new genotype? Okay, fine. This means that the genotype will yield the liters plus 400 liters. So what is happening, people, here? I want you to understand this one. What is happening, my people, here is that we have this allele. This allele, it is what? It is a dominant. This gene, it is what? It is a dominant gene. Earlier on, without this one, if iganje ignika ubani u-4000 bayabuya la u-4000 liters, then bayabuya la bathi once kwaba ne-dominant gene kuzo-edeka ubani u-400. Lo 400 le u-edeka kubani kulo 4000 that we already have. Ake sisibheke lana manje. Sesithini la sihlobo sami? Sesithi la nayi i-phenotype yethu esinayo. If we have this phenotype besivele sinobani sino 4000. Now we have what? A dominant gene. This dominant gene will add what? 400. It means it will be the 4000 plus the 400 that is added by the dominant gene, which is an additive gene. Then we get what? We get 4400. That's how we get it. That's how we get it. Okay, fine. Let me take it further to here. Now let's say, find the phenotype of this genotype. You see that here. Sorry. Now we are given. I'll have some difficulties. Here is our genotype. Here is our genotype. Here is our genotype. Yes, this is our genotype. They want us to find the phenotype. Remember we said earlier on, we already have 4000 liters. Then we said a dominant gene is equal to 400 liters of milk. Now what is happening? We have this genotype. So what is happening, people, here? It will be 4000 plus, this is a dominant gene. And yet, since it's a dominant gene, it means it will add how many liters? 400. Then we have 400. This one is a dominant gene. And yet, it means it will add 400. So we'll have plus 400. What about this one? It's a dominant gene. It adds a set of amount that is 400. What about this one? It's a recessive. It does not add. So it is a non-additive allele. This C, it's a recessive, a small letter. It does not add. The other C, it also does not add. So meaning, what will happen, people? We'll have 400 plus 400 plus 400 again, plus the already given 4000. Then we will have our answer, which will be very simple. We will have 4000 plus 400, 400 plus 400, then it gives us 5200 liters. These 400, it represents A, it represents what? B. That's how you get it. So for you to understand it in a simpler way, once you see a capital letter, it means there's a set of amount that is added. Then when I find the question yakubheka ukuthi but i-dominant gene noma i-additive gene i-adda how many, i-adda i-amount enganani. Maybe let's say, for example, let's say bayakunika bathi i-genotype ethile ikunika 60 eggs, ne? Then but a dominant gene, an additive gene will add 5 eggs. If but a dominant gene will add 5 eggs, kusho ukuthini lokho? Kusho ukuthi once kwaba ne-dominant gene kuzo-edeka u-5 eggs. Lo 5 eggs o-edeka on top of what? Of the 60 eggs that we have. So it means we'll have how many eggs? 65 eggs. I hope you get this one. I hope you get this one. Let me make it difficult for you. Let me make it difficult for you. Now the question changes. Find the genotype of this phenotype. Find the genotype of this phenotype. Yabo manje sesiyayishintsha. Sesikunika i-phenotype sithini? Thola i-genotype. Kanti ngale ngenhla sikunike ini? Sikunike i-genotype sathi thola ini? I-phenotype. Let's see how we get it. I will give you a very simple way of understanding this one, and I want you to understand it in a very, very easy way. How do we do this one, my people? When you get this question, the first thing that you must do, you will say the given phenotype minus the original phenotype. Senza njani la sihlobo sami? Le phenotype esinikwe yona la minus le phenotype le abasinike wena kule scenario abesinayo earlier on. So how do we do it, my people? So people here, we will say 60, we will say 6,400 minus 4,000. We'll say 6,400 minus 4,000. If we say 6,400 minus 4,000, what do we get? Let's see. We say 6,400 minus 4,000. What do we get? We will get 2,400. What do we do with the 2,400? We say the 2,400 divide by the amount that is added by a dominant allele. We say the answer that we get divide by the amount that is added by a dominant allele. Are we together, people? So when we say divide by the amount that is added by the dominant allele, we will say 2,400 divide by which are how many, what is the amount that is added by a dominant allele? It's 400. So we say 2,400 divided by 400. So then we do the division. We'll say 4 and 12, 16, 20, 24. So we will have the answer yet okubani oku 6. That is our answer. I hope you are wondering why we are doing this. Let's understand ukuthi lo 6 usitshela ukuthini ngane zakwethu. Lo 6 lo, le answer sithiwa lo 6 lo usitshela ukuthi ama-alleles, ama-genes a-dominant okufanele sibe nawo kufanele abe u-6. Kufanele abe u-6. Let me start this one afresh. You can even take your pen and write it down. You will write what I say. The question says, find the genotype of this phenotype that is 6,400 liters. thina sinikwe ukuthi sinikwe genotype yokuqala kulesa slide yaba 4000 sithi then now sebathi asitholeke manje genotype ya 6400. What do? Uthi lo 6400 minus lo 4000. Simple. Le phenotype abathi itholele genotype uthi yona minus le phenotype onikwe yona from the very first beginning, 6000 minus 6400 minus 4000 uthole ubani u2400 bese uthi lo 2400 uthi 2400 divide by the amount that is added by each dominant allele, then uthole answer. Le answer uyithola ikutshela ukuthi kufanele abe mangapi ama dominant gene. So la bathi 6 ne, if bathi 6 kusho ukuthini wena ama dominant gene kufanele abe mangapi, abe u6. So sozobe siyabuya nokuthi i-answer yami la kusho ukuthi ngizoba no A1 ngibe no A2 ngibe no B ngibe no B, there are 4 now, ngibe no C ngibe no C. Okusho ukuthi the first A contributes 400, the second, all of them will contribute 400, then these 400 are about 6 plus the 4000 they will give you 6400. Even if you can say 4000 plus 400 multiplied by 6 in a bracket is 6400. Let's see it in this way. It's 6000 minus 6400 minus 4000. It gives you what? 2400. Wenza njani ngalo 2400 lo? So to do 2400 divide by the given amount that is added by a dominant allele. Ignikubani u6. Lo 6 lo represent the number. Ngicela uyibambe la sihlobo sami. This 6 represent the number of dominant alleles you must have. Then as a result we will have such genotype. It's simple. 6000 minus 4000 thole answer yakho. Divide by the given number, by the amount that is added by each dominant allele. Leyo number itholayo ishothi kufanele abe ama alleles abengako. So it's simple in that way. Okay, fine. Let's look at it in this way. La I have given you a whole table. Siqale sathi sino 6 sine first genotype. This is your genotype, this is your phenotype. Ignigalo. Once I add the allele A1, a dominant, sesizothini 4000 plus, remember C10, a dominant gene, it will add 400. So sizothi 4000 plus 400 ignika ubani 4400. Now sesinama ngapi? Awu2. Ukusho ukuthi sesizothini manje? 4000 plus 400 plus another 400, it gives us 4800. But learners vele sathi 2 multiplied by 4, it gives you 800. Then bese sithi 800 plus 4000, it gives you 4.8. Coming here, let's do this one. Here we have how many? We have 4 wama dominant allele. Once waba na 4 wama dominant genes, it means sizo 400 abangapi? About 4. As we have here, sathole answer yethu. So that is the simplest part of it. Yabona siqeda ukuyenza manje this one. We had this, then thina senza njani sathi let divide minus 4000 sathole 2.4 divide by 400 sathola ama alleles awu 6. As you can see that we have 6 dominant alleles. That is the simplest part of it. Once kwaba ne dominant, yabona nje la kwaba ne dominant gene eta lo 400 sitshele ngawo. Kwaba ne dominant gene asawu2 lawo. Kusho ukuthi so eta ama 400 awu2. Lana sawu3 eta ama 400 amangapi? Awu3. That is the simplest part of it. Okay, fine. So it's very simple in that, man. We will take over sense in atavism. But before we do atavism, I will give you one calculation. Make sure that you do it. I will give you one calculation for me to see that you are getting it. I will give you that calculation. Try to do it. If you find it hard, you tell me. I will assist you. I will assist you. So make sure that you do that for me. Thank you, thank you, thank you. Tabetze here.